Hi, these are notes on evidence for evolution and mutations. So this is going to help give you an idea of how we knew that organisms were related in the past and how we determine that degree of relatedness now, and also the mechanism by which offspring of currently living organisms, so offspring of parents, can end up with changes in their DNA. So we're going to talk all about that. All right. So first of all, a long time ago, right, people didn't actually understand that different organisms were related. So they didn't understand that, say, a horse was actually related to a cow, or even larger, that a horse was related to an oak tree, or that a horse was related to a mushroom, or that a mushroom was related to a bacteria, which was related to a horse. So all of these things, we now kind of take for granted that they are related, but I want to go over a little bit of evidence for how we figured that out. So the first type of evidence we had was fossil evidence. Fossil evidence was simply being able to look at different strata. And you remember the law of superposition from 6th grade, that the strata on the bottom is older than the strata on the top. And so the strata on the top is younger. And this really was just evidence that organisms did change over time. If there were all these fossils of some sort of organism 530 million years ago, and then there are none 500 million years ago, that tends to indicate that these organisms have changed. Maybe they've changed into something else, maybe they've died out, but this was our first piece of evidence that things did in fact change. So then we started to look at skeletal structures. So we wanted to look at more underlying structures. And some of you know this intrinsically, that if you look, this is a human hand, a cat forelimb, a whale front flipper, and then this is a bat's wing, that they have the exact same skeletal structure underlying. So even though a hand and a flipper and a wing and even the forelimb of a cat do different things, have different purposes, that underlying bone structure is almost identical, which gives us evidence that those organisms are pretty closely related to each other. It can't just be random that there's all these similar underlying structures, especially when they have different functions, right? That tells us that the function is maybe connected to the environment, but the underlying structure is the same. So that's one type of skeletal structure. The other type that we use are what are called vestigial structures. So this is a human, obviously, and this though is a snake, which we don't think is very closely related. But if we look at a snake which has no noticeable legs, a snake is actually categorized as a quadruped, having 4 limbs. And that's because it has these vestigial hind limbs. Vestigial means no longer used, but the bones are actually still there, similar to our hind limbs. Now, these bones are not as similar as the cat. So we would say, yes, snakes, humans, and cats are all related, but humans and cats are more closely related than humans and snakes, or snakes and cats. So this works in your family as well, right? Obviously, all humans are the same species, but you could say, I'm related to my sister and my cousin, but I am more closely related to my sister than I am to my cousin. So when we're talking about relationship, realistically, we're all related, but I'm more closely related to my sister than I am to one of my 8th grade students, although from an evolutionary perspective, we are all related. And then the last piece that we used was embryo development. So here's a picture of different embryos in their various stages of development. And we can see the shark, the turtle, the chicken, the human. They all have a bony tail region, and they all have gill pouches at some stage in their embryological development. Now, we know humans aren't born with tails and gills, right? Chickens aren't born with gills. Neither, quite frankly, are turtles. Although turtles and sharks and chickens are all sort of born with tails. Chicken is a little bit of a stretch. But this shows if we all end up with the same development as embryos, there's got to be some connection there. There's got to be a reason that humans have gill pouches as an embryo when they're not born with them. Well, that gives us an indication that we're actually related to these other animals. And again, if we're now going back to the embryo, that's going to be less closely related, right? We're going to be more closely related to the snake, where we can see some of the skeletal structures, and even more closely related to, say, a whale, where our current skeletal structures are really similar. Then we are related to the shark. But this gave people the revolutionary idea that these organisms are actually related to each other. So let's just take, before we go into mutations, a brief review of DNA to traits, right? This is what we just finished. So remember, we have our DNA that's translated into RNA, or sorry, transcribed into RNA. It's translated into proteins, and those proteins are what lead to our traits. But that means if there's a change in the DNA, there could be a change in the proteins, which would then result in a change in the traits. And we've seen a little bit like, we've seen functional and non-functional proteins, right? A change in a protein can lead to a huge change in the trait. If now that protein isn't the correct shape for PTC to latch on, then you taste PTC as neutral. That's totally changed your trait. It could, for example, insert a stop codon too early. Then you get a smaller DNA strand. It could code, a smaller polypeptide strand. It could code for a different amino acid so that it's the same size protein, but now that's going to fold differently. And so you're going to get a different protein, because remember, the order of the amino acids is what leads to the shape of the protein, which is what leads to your traits. So there are three main types of mutations. So one is called a substitution mutation. And what that is, it is literally substituting an incorrect base for the correct one. And this would happen during the S phase of mitosis, and then your replicated strand would be different. And so then in meiosis, you would end up with a different DNA strand than you had originally, okay? So like here, if we had our initial strand as ATTGACGGA, right? The T latches on, the A latches on, but then it's just a mistake. Mistakes happen even in DNA. And so you get a C here, right? And then everything else is the same, but this has now changed from what should have been TAA to TAC. Now remember, when you're now reading that as mRNA, when the ribosome is reading it, that may or may not actually change the protein. So if you look, I just pulled this little bit off the codon chart, right? So all of these 4 codons actually code for threonine, or whatever it's called, threonine. I don't know. Anyway, but that means you could actually have a mutation, and because there's redundancy in the codon chart, it doesn't actually change the protein at all. The same things, this is isoleucine, that one I happen to know. The same thing if this last base changed here, but if it changed to a G, that would now change this would be methionine, that would change the amino acid. So that would be a bigger deal than one of these mutations. And that helps to explain why there's redundancy in the codon chart, right? It's not just to make life redundant, but that mutes the power of mutations, meaning everybody has mutations. Everybody has, right? DNA isn't perfect. Everybody has 30 to 40 changes in their DNA from their biological parents. But for a lot of those changes, they just pass unnoticed in the population. And that's because of this redundancy, that you could end up with a different codon and the same amino acid, so it's just the same protein. However, you can also get frameshift mutations. Frameshift mutations are either like a substitution mutation, which, insertion mutation, which is where one or more bases is inserted into the strand of DNA. So here we had A, G, T, A, C, C. So this would be the T, the C, the A, the T, but now there's another T that's pushed in there before the two Gs. So it's actually, if you think about a chromosome, right, that's so long, that's going to push everything down one, and that's going to really screw up all of those codons. It's not like a substitution mutation where it's just screwed up one codon, and that one codon may have a redundancy, so it might not screw it up at all. And then the opposite of an insertion mutation is a deletion mutation. So that's one or more bases is deleted from the strand. And so that again shifts everything down. And those of you who have read the book Wonder, a lot of us read that, it's a really good book. That one of the conditions that the boy in Wonder had was from a frameshift mutation. And so then you can see like a cascade of different genetic effects. And so then now we can bring it back to our evidence. So in today's world, what we use really, we don't look at I mean, we can still look at skeletal structures and we can look at embryo structures and things like that. But really what we look at is DNA. Now that we understand and have the ability to sequence DNA, really we're looking at how similar is the DNA. So if you take these 5 species, these are all mammals, they all have hemoglobin, but the sequence of amino acids that make up the hemoglobin in the different species is slightly different. Now, if they all make hemoglobin, this again gives us evidence that they come from a common ancestor, okay? Because it wouldn't be random that they would all make the same molecule to do the same function. There has to have been some ancient ancestor that that made hemoglobin, and then all of the, right, if here's our ancient ancestor, then all of these species branched off. But we can tell who's more closely related by how similar their hemoglobin molecules are. And so we can look at the amino acid sequences. And so then this is a comparison chart where we see if we're looking at the same part of the hemoglobin molecule, hemoglobin's huge. These are just 4 amino acids. I know we could look up how many amino acids are part of hemoglobin. My hunch is it's like over 1,000. But the human and the chimpanzee, this segment is identical. They have 0 differences. The human and the gorilla, or similarly, the gorilla and the chimpanzee, they have 1 difference. But if you compare that to like the human and the horse, there's actually 3 differences. This amino here, I'm gonna circle them here. Okay, this amino acid is different, this amino acid is different, and this amino acid is different. So that's 3 differences compared to 1 between human and gorilla, right? So I'm going to use the chimpanzee because we know chimpanzee and human are identical. Here, this is the same, this is the same, this is the same, this is different. So that tells us that yes, all these species are related, but humans and chimpanzees are most closely related. That means they split off from their common ancestor more recently than gorillas split off. Gorillas split off before humans and chimpanzees split off. Horses split off before gorillas split off. So horses, yes, were related, but that is a much more distant ancestor. So in today's world, what we use, now that we have accepted this understanding that, yeah, we're all related, now we want to look at how closely related are we? And that's what we can use our DNA evidence for. All right, if you have any questions, make sure you ask them in class. Have a really good day.